Welcome back. So we're we're in another long stretch of the course. The previous very long one, which took us like 3 weeks, had to do with dynamics. Life is dynamical and all that stuff. And now, I'm venturing forth the hypothesis that my physical biology, the cell co-authors, and I are now calling the real secret of life, which is defiance. So, so we've already talked about two kinds of defiance. We talked about defiance of fixed law, if you like. It's really the defiance of the second law at the end of the day. And in some sense, there is no defiance of the second law. So it's a bit of a wordplay because all that you're really doing is you're defying the second law in some local pocket or something, if you want to think of it in a little bit of a sloppy fashion. But at any rate, we defied fixed law in the sense that we didn't run down a concentration gradient, and then we defied Boltzmann and Langmuir. Maybe I would say we defied Langmuir in talking about adaptation. And my reason for saying that is that a dose response curve, cooperative or not, has some midpoint, and the midpoint is the midpoint. And the thing about adaptation is that we get to shift those midpoints. And this is something, that I have already mentioned. There's some really cool papers from Yu-Hai Tu and his collaborators and Ned Wingreen and his collaborators in which they really talk about this in some very, very nice and thoughtful ways. What we're going to talk about now is another kind of defiance, which I'm going to also cast as a defiance of Boltzmann, but this is the defiance that is associated with biological accuracy. And so, here I just remind you of the central dogma of Francis Crick. I love this thing on the left. Typically, it's very Crickian. If you go look around at the papers that he wrote by himself, they're very Crickian. They have a style. The guy had style. There's no other way to put it. And, here he's talking about the doctrine of the triad, which we now, call the central dogma. Once information has got into a protein, it can't get out again. And, we can have debates about whether or not he nailed that or whatever. It doesn't matter for my purposes. What I want to say. And by the way, I just want to make sure you see him at the bottom and also his little diagram of the central dogma. So we've already talked about the way that we extract biological information through the central dogma in the form of transcription. And we learned that process of transcription is regulated by both activators and repressors, as shown on the right-hand side of this diagram. But the thing I want to point out is that there's a different kind of regulation, if you like, and I'm going to refer that to that as specificity regulation. And what I mean by those words is that the cell takes care, and indeed even viruses take care. I'm amazed. Today is February 16th, 2021. We're living in the middle of an awful pandemic, which is just, it's claimed so many lives and disrupted economies and caused all sorts of mental health issues and struggles and whatever. And yet, I'm almost embarrassed to say it, but what an incredibly cool virus. The coronavirus, SARS-CoV-2, is so interesting. And one of the things that's interesting about it is it has proofreading, which is, in some sense, the topic of today's vignette. And if I have time and if I have knowledge and I have the right amount of effort on my own part, I would love to tell you about Manfred Eigen's idea of quasi-species and the era catastrophe, the argument that a certain virus genome, if it gets bigger than a certain amount, which is around 10 kb, there will be an error catastrophe where the very idea of maintaining oneself as a viral species doesn't hold water. That's a little too vague, but at any rate, so how many errors? So I'm going to play a little segment of this video, and maybe it'll be patchy, so I hope that that's not the case. Before I do, let me just remind you that DNA replication is a highly accurate process. And to put that in perspective, I want to appeal to this wonderful paper by Tanya Baker, which was in a wonderful issue of Cell at the turn of the millennium. And in it, she makes this wonderful analogy, that I think is very enlightening. And, I don't know how you feel, you the listener, but I personally am very persuaded by and find these kinds of everyday analogies to be helpful just to get a sense of a particular process. Like I just rode my bike across the Golden Gate Bridge yesterday, and the reason I bring that up is because I've always used this analogy of the packing of DNA into the viral capsid as being like taking 500 meters of Golden Gate Bridge cable and putting it in the back of a FedEx truck. That's the way we should think of. It's the right dimensions, it's the right stiffness, the right aspect ratio, and so on, to compare to a virus with its DNA. And I find that useful. It's useful to sort of think about the everyday scale. And so here, what Tanya Baker had to say is that DNA replication, we can think of this as the nucleotide express. If you have a FedEx truck traveling at let's say 500 kilometers per hour, or 600 is what she says. I think it might be closer to 500. And it makes a delivery of one of 4 types of packages on both sides of the street every 10 centimeters while traveling 500 kilometers per hour. Every 10 centimeters, a delivery of one of 4 boxes. And it makes its daily journey in 40 minutes and makes a delivery mistake once every 3 years. That's the accuracy of DNA replication. And yeah, it's like the flying over Greenland with the shades down. I just believe that, learning that factual thing, which is a biological numeracy thing, one has to, pinch oneself and just say, dude, do not take that for granted. Don't imagine that as some sort of just like, yeah, the error rate is 10 to the minus 9 per base pair per replication or something like that. No, I cannot stomach that. So, here's the process. If I hope the video works.
Using computer animation based on molecular research, we are able to picture how DNA is replicated in living cells. You're looking at an assembly line of miniature biochemical machines that are pulling apart the DNA double helix and cranking out.
So I'm worried that the video just didn't work well enough. But so what I want to say is that in this picture, from the left is the DNA that's being replicated. It's split by as she pointed out, the helicase, which opens up the DNA double helix. And then because of the 5' to 3' copying regulation, then in one of the directions, the DNA synthesis is continuous, and in the other strand, you have to copy backwards. So this leads to this whole story of looping and Okazaki fragments and that kind of thing, which is amazing and incredibly cool, and a great triumph of modern biology. It's kind of stunning that we even know such things, but okay, we do. And all that I'm trying to say is that that machine is moving along at 500, in everyday scales. If I make DNA that at 1 diameter in 1 meter diameter, then in that scale, the replication machinery will be of the order of the size of a FedEx truck traveling at 500 kilometers per hour, making a delivery on both sides of the street every 10 centimeters, making a mistake once every 3 years. Okay, so that's the deal. And we can put together a summary of the error rates in translation and transcription and in replication. And that's cool. I mentioned earlier that numeracy changes conversations. It changes the conversation to know that an action potential travels at 30 meters per second. It changes the conversation to know the age of the Earth, even though I would claim, really in some sense following Daniel Fisher, I would claim that we don't know how to reconcile the age of the Earth and biological evolutionary timescales. The reason I say that is can anybody, on the basis of even an approximate argument, tell me how long it takes to evolve a whale after the dinosaur slayer asteroid? And I think the answer to that is no. And in 10 million years, you go from mammals this big to 10 meters long or so. So at any rate, the numbers change the conversation, and here the conversation changer is shown on the lower left, where you see that the error rates are like 10 to the minus 10 per base pair per replication. It's really astonishing. But even the translation rate errors are much lower than you would expect. And so now this is one of those moments where I'm favorable to the idea of surprise, because as I quoted to you from the Abraham Peiss book about Einstein, in order to feel surprise, one has to have a theory or a prejudice. And so my prejudice would be that the first thing I learned about translation was about codon anticodon recognition, that you have triplets and that they meet each other And that immediately suggests a model of specificity. And the model is an equilibrium model in which we imagine, let's see, we imagine, well, okay, so what I want you to do is I'm going to try to make a simplified universe in which there's only two kinds of amino acids, either hydrophobic or polar. And in that world, which is naive, then protein folding, we're going to imagine protein folding is the segregation of those parts that are hydrophobic and the external exposure of those parts that are polar. So that's what we mean by this so-called HP model, hydrophobic polar model. So in that world, there's really only two tRNAs. And so what I'm going to do is I'm interested in the question of the error rate in this very naive two-state picture of translation, where what I'm going to do is I'm going to imagine a states and weights diagram like we've done many times over the course of this, this series of vignettes. I'm showing you in the top, it's a little bit subtle, but notice the mRNA. So the mRNA is shown as the little thing at the bottom in the center. And now we have correct and incorrect tRNAs competing for the affections of that mRNA, so to speak. And what we want to know is what's the relative probability of binding the wrong versus the right. And so the probability of binding is given by the concentration. So the statistical weights, I guess, let me start with that, shown in the right column. I'm just using states and weights in the ways that we usually have. So I'm saying that the thermodynamic weight of the mRNA when it's empty, I'm calling that 1. That's basically my zero of energy. And then, as we've done many times, the statistical weight for binding is going to be concentration divided by the KD. So here I say that the concentration of the right substrate is the right tRNA is CR. The concentration of the wrong substrate is CW. If CR and CW are equal in concentration, then P, so I want to know the probability of the state where I have the correct tRNA on the mRNA, and then I want to know the probability that I have the incorrect one. And the ratio of those two probabilities is going to be given by K wrong over K right. And that's it. Fidelity comes down in this equilibrium mindset to the ratio of the KDs. And I'll probably have everyone think that through. So Linus Pauling actually was thinking about this long before people like Hopfield and Ninio, and before Pauling was Schrödinger in his What is Life. He was already worried about these questions. He came at it from a statistical physics point of view, which I think I mentioned at the beginning of the term, and I love that very much because he was, schooled in the Boltzmann school. He knew about the square root of n fluctuations. He wanted to know how big a gene was, and the reason is because he was in this mindset of saying, well, how big is the gene? The reason I want to know that is I want to know how it is that genes could be so stable. You could make the argument, I don't know if you all will buy this, but you could make the argument that in some sense the Hox genes are more stable than the Earth. And the reason I say that is, 50 million years ago, India collided into Asia, creating the Himalaya mountains. Those are 8,000 meter mountains. And in the same time, the Hox genes have more or less, roughly stayed the same. You can recognize them. Anyway, okay, never mind. So Schrödinger was thinking about the fidelity problem. Pauling was definitely thinking about this. And what Pauling said was, well, hold on, if the wrong codon-anticodon pairing is a matter of a couple of hydrogen bonds not being correct, then that tells me the energy scale, or another way to say it is it tells me the ratio of those KDs. And so I can compute using Boltzmann statistics. I will offer this as a homework for people to think about. So let me actually make a note to this effect so I don't forget. So just bear with me for a sec. I'm forgetful, and so I definitely need to put this on my list. So equilibrium fidelity. All right, so what I'm doing here is I'm basically using Boltzmann statistics, and what I find is that, remember, a hydrogen bond is of order a couple of KTs. E to the 2.3 is 10. So for every 10-fold improvement in accuracy, I got to acquire another 2.3 KTs worth of energy. So if I have a delta E of order 5 KTs, that's worth about 100 in specificity. So here I plot for you the error rate, the ratio of error to correct as a function of this delta epsilon. And this is only for my hydrophobic polar model. Remember, if I'm talking about actual ribosomal translation, there's way more wrong tRNAs around than there are right. There's not just two amino acids. There's many, many possible competitors, and all those competitors are vying for the attention of the codon. Um, so, so at any rate, this is the surprise coming from a wrong model. We have a wrong model. The wrong model leads to us to biological numeracy. We can't just talk about it. It's not a words topic. We have to actually do the math. We do the math of equilibrium binding, as shown here, and in so doing, it reveals to us this interesting observation that that number is very inconsistent with these observations of like a 1 in 10,000 error rate in translation. Okay, so that's the puzzle. That's the conundrum, and the answer to the conundrum, which I will talk about in the next vignette, was a beautiful set of papers that I think are highly transformative. Not everyone agrees. I've talked to some people that work on the structure of the ribosome that think that this work did more harm than good. So, you know, that's the nature of the scientific enterprise. Of course, you can try the H-index worldview, and you can try and find out how often these papers have been cited, and the answer is lots of times. But, you know, science is not a popularity contest, despite appearances to the contrary. Science is about what in 100 years people will still hold on to, you know, in my opinion. Like we're in the knowledge generation business. We want to have bankable ideas. And I suspect that the idea of paying free energy to improve specificity is here to stay. That's my speculation, you know, and that's my take. So I think that this particular idea that both Hopfield and Ninio put forth was a really, really wonderful idea. And what I will do is I will try in the next vignette to do the calculations. But let me just tell you the essence of it. I actually asked Hopfield about this. So I told him, you know, I imagine that you did this sort of Boltzmann estimate, and it came out wanting in the sense that the error rates were much better than you would get out of that simple Boltzmann estimate. And so then he complemented that with the insight that there was energy being expended. And he said to himself, you know, what's up with that energy expenditure? What does it purchase you? And what I'm going to show you when I try to do this formally, I'll show you this figure again. This is going to be my last figure of this particular vignette. What you see at the top and the bottom, let's go only to the first step, the so-called molecular recognition. So I have an enzyme shown in green. That enzyme can bind both the right and the wrong substrate, which I'm showing the right one is blue, the wrong one is red. And now that specificity, as I've already noted, is given by the ratio of the KDs. So that's basically this. So this e to the minus delta epsilon over kBt can be recast as the ratio of two KDs. So I have the ratio of the KDs. And so that gets me a certain level of specificity. Then, in keeping with my usual desire, you know, rather than showing you ATP hydrolysis, which is a bit of a magic wand, I want to either lift a weight or push a piston down or something like that to do work on the system that will then lead me to a new configuration and it will give me another chance at specificity. So here's the concept. The way I'm going to think of this is that I have this piston and I have a yellow effector ligand, which when it binds onto the enzyme, it changes its conformation and makes it now competent. It shifts it from the inactive to the active conformation. When it's competent, it can now perform its enzymatic action. And the point is that there's a certain rate constant for doing the reaction, which is the rate constant R, which means there's a waiting time 1 over R before you actually do the enzyme action, on average. Now the thing is that the off rates, because the KDs are different, the off rates of the right and the wrong substrate are different. The blue one has a lower off rate than the red one, and what that means is that more of the red ones will fall off prior to the enzyme action than blue ones. So we basically get a second try at rejecting the wrong substrate, and that buys us an extra piece of specificity, as shown on the right. In other words, I'm giving you an approximate formula which tells us that the specificity is the ratio of the KDs squared. This is like the simplest toy model of the Hopfield worldview, But there's much more to it, and people have generalized it in all sorts of interesting ways. And I will also comment on that as we proceed.